I'm sure you know what this is. Yeast is a eukaryotic fungus used in cooking and brewery, but did you know it's also an important organism for the biotech and pharmaceutical industry? This is due to its fast growth rate and simple media requirements, whilst also being able to produce more complex proteins than your standard bacterial strain. But what actually makes this organism so useful, and what are the underlying genetic mechanisms that allows it to behave in this way? Saccharomyces cerevisiae, or baker's yeast, contains many well-characterized core promoters, which are found just slightly upstream important genes. These are controlled by transcription factors, which switch these genes on and off based on specific signals or environmental conditions, such as the presence of glucose. Let's take a closer look at the plasma membrane. It features two specialized glucose-sensing proteins, RGT2, a high glucose sensor, and SNF3, a low glucose sensor. Together, these sensors enable the yeast to precisely detect and respond to various extracellular glucose levels. RGT2 and SNF3 initiate a signaling cascade that leads to the phosphorylation of adapter proteins, most importantly, YCK1 and YCK2. Although the precise molecular mechanism remains unclear, evidence suggests that the phosphorylation of YCK1 and 2 propagates the signal by targeting and phosphorylating downstream proteins. This process eventually leads to the activation of the SCF complex, a multi-protein complex crucial for the targeting of specific proteins for degradation. YCK1 and 2 are key players in this activation, marking a key step in this cascade. Keep this in mind as it will be important for later. Another consequence of glucose detection is the degradation of the transcription factor RGT1. As a repressor protein, RGT1 binds to the promoter sequences of hexose transporter genes, recruiting additional proteins to inhibit their expression. Structurally, RGT1 contains 3 zinc finger domains, arranged in tandem. Each zinc finger recognizes a specific 3-base pair sequence, with a 1-base overlap. In this case, RGT1 will recognize the CGG-AAAA motif found on the HXT1 core promoter. In the presence of glucose, YCK1 and YCK2 indirectly phosphorylate RGT1, causing it to release from the promoter regions of hexose transporter genes. Once phosphorylated, RGT1 is recognized and ubiquitinated by the SCF complex, which marks it for degradation by the proteasome. A similar mechanism involving MIG2 activates hexokinase genes, leading to the production of more hexokinase enzymes, which catalyze the first critical step of glucose metabolism. These mechanisms provide baker's yeast with a precise genetic and biochemical framework to rapidly regulate gene expression. But this doesn't really tell the full story. While gene-specific transcription factors and molecular mechanisms explain how these processes occur, it doesn't explain why these processes happen so quickly. Yeast achieves this rapid response through complex post-transcriptional mechanisms that regulate mRNA stability and degradation. For example, the mRNA encoding glucose transporters is stabilized in the presence of glucose by RNA binding proteins. While the specific glucose-responsive RBPs are still being characterized, it is likely that proteins such as PUB1 play a role. These RBPs respond to nutrient signaling molecules as part of a larger regulatory network. This stabilization increases the presence of the protective poly-A tail on mRNA, which enhances translational efficiency. The result is a higher rate of glucose transporter synthesis, boosting glucose uptake. Yeast cells also rely on specialized cellular structures known as P-bodies to regulate gene expression. Unlike traditional organelles, P-bodies are non-membrane bound structures formed through liquid-liquid phase separation. Within these concentrated droplets, intrinsically disordered regions of mRNAs, RNA binding proteins, and enzymes interact, storing important mRNAs for future use. In response to specific biochemical signals, these droplets dissolve, releasing stored mRNAs related to glucose transporters and hexokinases. This rapid release enables swift translation by ribosomes. All of these cellular mechanisms allows yeast to rapidly adapt its metabolism in response to glucose. This remarkable adaptability is crucial for biotechnology and pharmaceuticals. It boosts the cost effectiveness and efficiency of production processes, resulting in high yields of recombinant proteins and life-saving pharmaceutical treatments. And of course, yeast has been essential for thousands of years, helping create the delicious food we enjoy every day. I hope you've enjoyed this short video about yeast gene expression, and thanks for watching.